All right, guys, topic 6.8 is on biotechnology. So the big idea here is that all this DNA that we have been talking about and we've been learning about, you can actually manipulate the DNA using some biotech methods. And that, of course, would give you different proteins that are produced, different genes that could be expressed. So we're going to talk about the first 3 sections of notes in this lecture. And then tomorrow you'll come back for the last section for DNA fingerprinting. Yes, you'll all come. Yes. Okay, so first, let's talk about something called a restriction enzyme. So we'll start with what is a restriction enzyme and not watch that video. Restriction enzymes are enzymes that are specifically designed to make cuts in DNA. So they're going to basically chop up DNA into pieces. Now, these restriction enzymes are found in nature in bacterial cells. And it's actually thought that it's a part of the bacterial cell's immune system to be able to cut out segments of DNA that are not helpful, not beneficial, making them sick, have been inserted or mutated. And so we have sort of understood what these restriction enzymes do naturally in bacterial cells, and we have pulled them and now find a use for them in several biotech processes. Because they're found in bacterial cells, the names of these restriction enzymes are named for the bacteria that they're found in. So one great example of a restriction enzyme that we see repeatedly is EcoR1, and so that comes from naturally occurring E. coli. Restriction enzymes will essentially scan a segment of DNA, and they're looking for sequences, specific patterns in the DNA to know where they're going to make cuts. And when they find those patterns, they make a cut. And so these sequences are called restriction sites. And so this EcoR1, this bacterial enzyme found in E. coli, would make a cut at restriction site 1 in that particular E. coli's bacterial sequence. So when the enzyme makes a cut, we're going to see a video here, a really short video. Notice in the video that when the enzyme makes a cut, it can make something called a blunt cut, or it can make a cut that produces these jagged little ends, which are called sticky ends. So let's check out the video. Maybe.
Restriction enzymes cut specific recognition sequences in DNA. The enzyme EcoR1 recognizes the sequence GAA-TTC and produces a staggered cut with 5' single-stranded overhangs. The enzyme PST1 recognizes the sequence CTG-CAG and produces a staggered cut with 3' single-stranded overhangs. The enzyme SMA1 recognizes the sequence CCC-GGG and produces blunt ends on the DNA.
Okay, so you could see the 2 different possible kind of end results of the DNA. They're either blunt cuts or they produce staggered cuts, which give us these things called sticky ends. So restriction enzymes essentially will cut DNA into fragments. And now we can move on to how we could possibly use restriction enzymes. And so using restriction enzymes, we'll talk first about how we can use them within their sort of natural habitat of a bacterial cell, and then how we could use them in our own genome. So for bacterial cells, we can use restriction enzymes to create plasmids that we want. We can make our own plasmids and give those plasmids back to a bacterial cell and make that bacterial cell produce something that we want. Very much like our pea glow lab, where we were inducing the bacterial cells to glow because of the plasmid that they had taken in, which had been bioengineered. And so if we're looking at how would you do this, it's a pretty simple step through process. You would first use restriction enzymes to cut out a desired gene from a segment of DNA from an organism. So, for example, the actual GFP, the green fluorescent protein gene, was cut out of a genome that was a jellyfish genome. Then you would use the restriction enzymes to cut open a plasmid, and then you would insert that new gene into a plasmid. Now, we did it in the lab, but this is also being used in lots of unique ways, one of which is producing insulin. So if you know anything about people that are diabetic, there's two types of diabetes. And one type, you're insulin dependent. You have to take insulin as an injection. And so producing that insulin, a long time ago, we used to harvest it from other animals, from pigs. Now we can produce human insulin using bacterial cells. And so check out this.
We'll use the example of how the insulin gene is inserted into a bacterium in order to manufacture large amounts of human insulin. The gene that codes for human insulin is found on chromosome 11 at position 15.5. The insulin gene is cut from chromosome 11 using special enzymes called restriction enzymes. Bacteria possesses small circular strands of DNA floating in their cytoplasm. These strands of DNA are called plasmids. A plasmid is extracted from the bacterium cell. The plasmid is cut open using the same restriction enzymes that were used to cut the insulin gene from the human chromosome. The restriction enzymes leave sticky ends, where one of the two DNA strands is slightly longer than the other. Because the same restriction enzyme was used to cut both the human DNA and the bacterial plasmid, the sticky ends are complementary and will allow for joining using complementary base pairing. The insulin gene is joined to the plasmid by the sticky ends. An enzyme called ligase is used to complete the joining of the two pieces of DNA. The modified plasmid is then reinserted into the bacterial cell. The modified bacterium is a genetically modified or transgenic organism because it contains some human DNA along with its own bacterial DNA. Its bacterial DNA is called recombinant DNA. Bacterial DNA recombined with human DNA. The bacterial cell is placed in a fermenter, which allows for rapid asexual reproduction in ideal conditions with the optimal temperature, pH, and lots of food. Because the bacteria reproduce asexually, the offspring are all clones of the original transgenic bacterium. They all have identical recombinant DNA. The bacteria express their own genes in order to survive. In other words, they make their normal bacterial proteins. But they also express the human insulin gene and make human insulin. The fermenter has become a culture of mini factories, all producing human insulin. The insulin is then extracted, purified, and packaged. Humans can now inject themselves with human insulin. And that is how genetic engineering works.
So a really easy example to see how creating a plasmid using restriction enzymes can benefit us, right? So pharmaceutical companies producing insulin, we can now utilize bacterial cells to make that insulin and then extract it and purify it. And we have human-based, right? Because it was a human piece of DNA that was inserted into that plasmid, we have human-based insulin instead of having to harvest it from other similar like animals like pigs. Oh, no.
We'll use the
Stop that. Okay, so another way that we could use restriction enzymes is in a process called PCR and also in DNA fingerprinting, which we're making the transition into how would we use this on human genomes specifically. And that is in the very next section of notes. So this really just says human DNA and PCR and DNA fingerprinting, see section below. Let's talk PCR in the section below. So there is a technique called PCR, polymerase chain reaction. And really what we're doing in performing PCR is we are taking a very small sample of DNA and we are increasing the amount of that DNA in a sample so that we can run a test on it. So, for example, maybe at a crime scene, there's a very small amount of blood that has been left behind, and that small amount of blood is not enough to run testing on. So we would need to amplify or increase that sample so that we could actually run tests on it. Now, PCR is something that's talked about right now a lot with COVID. The PCR test, if you go get a COVID test, one of the types of COVID detection tests is a PCR test. And so they'll take a nasal swab, right? If you've had these tests done, they stick a big swab up your nose, and they're hoping to capture a little bit of viral DNA in that swab. It's such a small amount that they run it through a PCR to boost the sample, and then they test for its presence. So PCR tests are kind of the gold standard right now for detecting COVID because it will actually detect the DNA of the COVID virus. In the process of PCR, you need a couple of things to be able to amplify a sample using this technique. So you're going to need a sample DNA, so a piece of template DNA. You're going to need some RNA primers. You're going to need enzymes that are going to help make more copies of that DNA because we're just trying to boost the amount of the original sample. Now, the most common enzyme that's used in PCR is called Taq polymerase. Taq polymerase or Taq poly is an enzyme that is found in, here's its protein structure. It's an enzyme that's found in a bacteria called Thermus aquaticus. And Thermus aquaticus, as a bacteria, lives in these hot springs. And so the benefit of using Taq poly means that we get to use an enzyme that comes from a bacteria that naturally lives in a really hot environment. So this particular enzyme can survive high temperatures, which is critical to the process of PCR. We're going to raise the temperature, lower the temperature, raise the temperature, lower the temperature. We need enzymes that can survive that temperature increase. We also need new nucleotides if we're going to create some DNA. We need some nucleotides to do that. And then, of course, we need the PCR equipment. So this is not something that this task is not something that you can perform without this specified bit of equipment. In running PCR, so if you have all of those ingredients, how do you actually run a PCR test? You are going to go through 3 different cycles of copying the DNA, and the cycles are all the same. So let's talk about cycle 1. Right, the process in cycle 1, we're going to take our sample of DNA and we're going to heat it up. And heating it up is going to denature the hydrogen bonds between the DNA, and so it's going to open the DNA up. And once the DNA is open, it will serve as a template for a complementary strand. And so we'll cool it down. After we've heated it up, we bring that temperature back down, and then we're going to do something called extension. And so we're going to make a complementary side of the DNA using the primers and the new nucleotides and the Taq poly. And so we'll do it once, and then we do the exact same thing a second time, and we do the exact same thing a third time. So here is an animation of that process, and you can see after you've cycled 3 different times, you're amplifying the amount of DNA that exists from beginning to end. So check this out.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
So you can just keep running PCR depending on how big your initial sample was. You can run it as many cycles as you want to get the amount that you would need for these tests. So here is a picture of what we just talked about. Here's our initial sample. Here are the primers, the new nucleotides, and then that Taq polymerase. So we're going to within the first cycle, we're going to heat it up, which denatures, so it's going to open up. Then we're going to begin the process of annealing, which is cooling it down, adding the RNA primers, and then extending. So you started with 1 piece, and at the end of cycle 1, you have 2. Well, then you're going to heat it up again, do the same process, and each of these 2 are going to open up, and 2 becomes 4, and 4 becomes 8, and it can just keep going depending on how large of a sample you need. All right, that is the section of notes for today. The next section of biotech notes will finish out this really short notes packet. So I'm going to talk to you about something called DNA fingerprinting, which can use the PCR amplified sample and actually run a test on it that is helpful for lots of different things. So we'll talk about DNA fingerprinting, and then we'll talk really quickly about CRISPR as well. So you are done with your notes for today.